Hey, thanks for having me. It looks like a really fun day. Great speakers and a nice turnout. And yeah, congrats on your center. I'm Tim from Oxford and UCL, and this is a mouse. And this mouse has learned to go to 4 places in a row to get to a reward. So here he goes to A. He's gonna go to A quickly. I'm going to zoom it through a bit faster than he can walk. So he can go to A and then B and then C and then D up here, and then he's going to go back to A this way, right? And so he's learned to do that, and so he can do it really well. He's done it 100 times or something, right? This is a mouse that's learned a sequence of 4 things: A, B, C, D. He has to go in order because the reward doesn't appear unless he's got the other rewards first, right? So he has to go A, B, C, D, A, B, C, D, A, B, C, D, right? Here he is again, but this is the first time he's ever seen this set of rewards in these locations. Previously, he's seen the structure A, B, C, D, the same kind of task, but with the rewards in different locations. And he's going to have to figure out where the new rewards are in this world. Here he goes, and you can see he's gonna. I'm just gonna cut in just before he finds A. So he's gonna find A, but then he's not going to be able to find B. He's going to go the wrong way first, and then he finds it, and then he's going to go the wrong way to C, and then he finds it, and then he's going to go really the wrong way to D. He doesn't know. He can't know where these things are, but there's one thing he can know. He can know that after he's been to D, he should go back to A because he's found A already. So here he goes, he's at D now, and now he's going to go back to A as fast as he can by the shortest possible route. He remembers where A is, despite the fact he's only seen it once. Right, that's called a zero-shot inference because he has never made that transition before between D and A, and the only way he can make that transition is if he understands something about the abstract structure of the problem that he's solving. This like A, B, C, D, A, B, C, D. When I've got to D, go back to A structure, right? Okay, so this talk is oh yeah, and so the other thing he has to do 2 things, right? In order to solve this problem, he has to be able to keep track of that abstract structure, A, B, C, D. And the other thing he has to be able to do is navigate in space. And so this talk is kind of arguing that these 2 things are the same. They can be done with the same mechanisms, and this realization lets us understand problems with lots of different, more complicated structures. That's basically the talk. And so now you can all go home. But if you want to hear a bit more about it, then we can just start with space. And here's a mouse solving what might be an easier problem, which is he walks along this tortuous route and finds some cheese. And then the next time he has to figure out how to find that cheese again. Either he can do that by taking the same actions again, or if he understands something about the structure of the problem he's working with, if he understands how space works, then he can do something like this, which is an awful lot easier. This is an inference, and this inference relies on some prior structural knowledge, which is the knowledge of how 2D Euclidean geometry works. Okay, awesome. So we know a little bit about how that, oh, sorry, there's a question in the chat. So I'm just going to leave the chat, and if you want to interrupt me, or I'll deal with them at the end. Okay, so we know a little bit about how this structural inference works in the brain. I guess many of you here know this. There are these cells which mediate these kinds of structural inference. In hippocampus, there are these things called place cells, which fire at particular places in a open, this is a square box, and this fires wherever the rat's here. And then there are these really exciting grid cells, which are in the entorhinal cortex, the medial entorhinal cortex, just next to here in the hip, this blue area in a human. Actually, these are actually from a rodent, so but there's an equivalent place, which fire on these hexagons. They also fire in these discrete places, but these places are on a grid, right? This beautiful grid, right? Okay, and alongside these two Nobel Prize winning cells come a whole buffet of cells which are not quite as celebrated, but all have their own nature papers, like object vector cells, border cells, landmark cells, etc., which all seem to be part of the same kind of structural inference problem, but bespoke to space, right? And there's one more thing that I need to tell you so that you can understand things coming up, which is in a human brain, at least, these grid cells are not restricted to the medial entorhinal cortex. They also happen in this network of association cortex in the medial frontal and medial parietal cortices. Right, here are some that are recorded extracellularly by Josh Jacobs. Excellent. Okay.
Tim.
Yeah.
I think people do want to find out a little bit more about that behavioral paradigm of yours. How long did it take the animal to learn that? And
yeah.
What was the actual?
Yeah.
Are you going to tell us more about that?
So far, I've only used that behavioral paradigm to illustrate the problem that I'm solving. But later on in the talk, I'll actually show you some data from it.
Okay, so you'll tell us more about the details.
Well, actually, I can tell you more about the behavior now. It takes, it's remarkable. It takes 5 or 6 structures for them to learn. So learning the ABCD, the first set of 4 ports takes them, in the very first structure, it takes them quite a long time, like maybe 200-300 trials before they're performing reliably, running 4 in a row. So that, I mean, in fact, more than that, sorry, yeah, hundreds of trials, 600-700 trials before they're performing 4 in a row. But you show them a few different examples of it and they quickly start to make the inference. So on the 5th example, they're already above chance of that D to A inference, way faster than any deep network would ever be able to do generalization. The other rather intriguing thing, I think, is that there's some hint that they bend the inference out so that they start off going A, B, C, A, and it's like they have a sort of circle and they sort of stretch it out. They realize they're doing the same thing again, and that repeating structure is something that they can learn from, and they do it quickly within 4 or 5 examples. Yeah, exactly. Does that answer your question?
Yeah, the other question was well, the other question was asking general things, some of which you've addressed, but I guess they just simply get rewards when they get to the right next port, right? So it's all trial and
there's
no queuing of the locations or anything like that.
No queuing. They just get the rewards when they get to the next location. Yeah, exactly.
Okay. And the person asks, is there any way to make the learning faster? But maybe you'll tell us about that as we go along. So I should let you continue. I'm slightly, it's only a half-hour talk and I've got 3 studies to get through. Okay. So good. We'll let you continue then.
But it doesn't matter, actually it doesn't matter. This is just talking's fine as well. I don't mind if I don't get through the studies. Cool, okay, well, let me just get into the argument that I was in the middle of getting into, which is that these cells seem to support spatial structural inferences, but the same system is is also involved in other kinds of structural inferences. Here's a really famous one studied by Howard Eichenbaum called transitive inference, which goes as follows. If I tell you that Jane beats up Bob and that Bob can beat up Alice, then you're pretty confident that you know who's going to win a fight between Jane and Alice, because you understand the nature, the 1D nature of the sort of greater than or better than operator. That works in rodents as well. If I tell you that A is more rewarded than B and B is more rewarded than C, then you can tell me, or the mouse can tell me, or the rat can tell me that A is going to be more rewarded than C. They'll choose A over C. And that also relies on the hippocampal formation. Believe me, with the fMRI, I'll explain the rodent lesion data. this says that if you make a lesion either to the entorhinal cortex, or which removes most of the sensory input from hippocampus, or to the fornix, which removes the frontal input to hippocampus, which disconnects frontal cortex from hippocampus, then rodents can make these A-B decisions, which are the ones you train them on, but never make that inference. The BD one here, this one here, can't do that after disconnecting hippocampus from the rest of the brain. Okay, good. And not only is it the case that the same brain area seems to be involved in these things, but also some of the same cellular patterns seem to be involved. Here's a famous paper by Dmitry Aronov, where he has, in David Tank's lab, found place cells, but these place cells are not for positions in space, they're for positions in frequency in an auditory task, right? So this animal is listening to sounds going whoop, which is like running down a 1D frequency space spectrum, like running on a 1D track, but now the track is made of different frequencies. And there are place cells for there. And at roughly the same time, we were finding something that looked like a smoke signal of grid cells for two-dimensional spaces, which were a long way from physical space. So this is the space. The 2 dimensions here are the lengths of the necks and the legs of birds, and so you can see something that looks like a hexagonal signal in that abstract space. Okay, so there's something about this structural inference that seems to require these kinds of brain areas and these kinds of cellular representations. So we started thinking about how to model this, and these are the kind of puzzles that we wanted to solve to start with. I'm just going to highlight James Whittington here because he's now at Stanford, just started with Surya Ganguly. And the kind of questions he was interested in asking are, how can this relational inference work? Can the same system solve spatial and non-spatial problems? And why do these cells look like they do? Okay, so when tackled with those problems, we always like to think we always like to take a step back and think, what problem is the brain trying to solve? And our take on it is that this is really the problem that the brain is trying to solve. It's like this. If I tell you that Janice has a brother, Bob, and a child, Alice, then you, because you're clever humans, can tell me all sorts of other things about that family. You can tell me, for example, that Bob is Alice's uncle. And not only can you do that, but you can learn that kind of relational structure in one family, and then you can immediately generalize it to any other family that you meet, and you can effectively path integrate your way around the relational structure in order to work out what the relationships, the other relationships are. So you have some abstraction, some abstract understanding of the structure of relationships and how they themselves relate to each other. So that's I think the essence of the problem that we're trying to solve, but it's not very concrete, right? How could you build a system that would do that, right? And so we can make that a little bit more concrete by asking this kind of question. Predict what comes next, right? If I tell you light bulb, right, broomstick, down, motorbike, right, etc., then what comes next? And nobody can solve that problem amongst the audience unless they've got extremely good spatial sort of rotational abilities. But you can solve that problem if you've seen loads of similar examples before that have the same kind of structure, in this case 2D space. Then you can abstract this relational structure from the sensory environment, and now you can just you can lay down the new sensory objects on this environment, and you can say, I can predict that the light bulb will come next. And so that suggests to us a couple of things. One is that this is really an abstraction and binding problem, right? I have to abstract something which is not an object now, but it is a structure, the structure of the problem you're solving. And secondly, maybe you could learn that. You could learn that by just predicting the sensory input and learning to do so fast, right? That's essentially how you can go about solving this kind of problem. All right, I just wanted to take a quick side alley to convince you that it's not that these kinds of ideas are not totally alien to the hippocampal formation, because I was talking about abstracting relational structure and binding it with sensory input. And here's a diagram that Howard Eichenbaum drew of the hippocampal formation, looking at the functional properties of the inputs. Here's the lateral entorhinal area, which codes for things like sensory input. Here's the medial entorhinal area, which Howard used to call context, but I'm going to try and persuade you is this like location in a coordinate space, which tells you the relational geometry. And then Howard would say, well, like in the hippocampus, we'll bind these things together. We'll do what to form a relational memory, a thing in context memory. All right, so we can just take that and build that model on the model, which is just exactly trying to build what was in Howard's mind, with the idea that we can use prediction to do the abstraction. Okay, and so here's this model. This is like this thing, the medial entorhinal cortex. Where am I in this weird, wacky coordinate system, this abstract relational space? This is the sensory input, motorbike. And then this is the hippocampus. It's going to say, it's going to bind these two representations together with an attractor network and say, okay, the motorbike is my uncle or something like that. And so we know what that looks like, and we're going to try and learn what that looks like by predicting, just like I showed you before with the 2D space, predicting what's going to come next. And the question is, can we learn interesting representations up here using these principles? Yeah, we're going to train it to predict what's going to come next. This is an interesting slide, actually, because it shows you really why it needs to understand the structure of the problem, right? Because it can go just like the mouse did. It can take a tortuous route through a world, and if it understands the structure of the problem, then even though it's never seen any of these gray lines, it knows all of those gray lines, even though it's only ever seen the red line, right? If you train it well, right? It can infer all of the links it's never seen. Okay, I could give you all sorts of details about the training of that, but I'm not going to, because I want to get onto some data. And so I'm just gonna, oh yeah, we're gonna learn those words. I'm just gonna show you what the representations look like if you do that. And you can guess what I'm going to say, which is in these abstractions, if you just do that and train it to predict the next thing, you get things like the medial entorhinal abstractions, right? So you get the grid cells. So here are grid cells in the model, which come at different phases. They also come at different frequencies. Here are real grid cells, but you also get a lot of these other abstractions, these structural abstractions that seem important for training space. So these border cells, these object vector cells that I didn't describe in great detail, but are other properties of spatial representations in the medial entorhinal cortex that generalize, that they're abstracted, right? They generalize across different objects with the same quality. So this object vector cell generalizes to all different objects. Okay, so these representations that are famously parts of the in the entorhinal cortex that are famously involved in encoding space are really just what happens if you try and do good sequence prediction and abstraction, and you happen to be working in a two-dimensional world. Okay, if you go and look in the hippocampus now, you can see things that look a little bit like the hippocampal representations. Here are some place cells. In the model, there were small ones and big ones. You could have guessed that would have been the case once you knew there were small and big grid cells, because these hippocampal cells are just binding the grid cells to particular sensory elements. And so any grid cell that has a peak at the correct sensory element is going to cause a place field in the place cells, right? And so that's an explanation of why place cells have this have a single peak, but grid cells have multiple peaks, because in the place cells you're being picked out not just by the grid, but also by the sensory stuff that's at that place. And you can see a similar effect here in the landmark cells in hippocampus, which care about how far away you are from an object. They don't care about all objects, unlike the entorhinal ones I just showed you. They just care about objects that have the right sensory properties. Cool. Okay, so that shows you that these models can do a good job of predicting these entorhinal representations, just thinking about abstraction and sequence prediction, right? But they can also, because they just think about abstraction and sequence prediction, and they aren't made in a bespoke way for 2D space, they can also model things that are not just spatial reasoning tasks. So here's one example of many we've tried now. I'm just going to show you a couple for time reasons, but this is a fun one. It's a mouse that runs around a lap 4 times and then gets a reward on the 4th time. And so if you look at what TEM is going to predict, the model is going to predict in this situation, it's going to predict that you're going to have some place cells. You need some place cells because you have to predict the immediate sensory thing that's coming right now. But you have to do more than that, because if you want to be able to predict when the cheese comes, you've also got to keep track of which lap you're on and where you are in that lap. And so you need another class of cells here, which are also a bit like place cells, but they only fire on one lap. They're like lap-specific cells. You need those. If you don't have those, you can never remember when the cheese is coming. Okay, you actually need a 3rd class of cell, actually, these counting cells, which increase slowly through the laps, so that these lap-specific cells know when to transition. So, so that's what these models predict you're going to find in these things, and that's exactly what you find if you go and look in the hippocampus, right? So here are these place cells that fire on every lap. These spatial cells, these ones, so these lap-specific cells, these ones happen to be firing on the 1st lap, and then there are a bunch of cells which appear to count slowly and grow as you go through the thing. And you could argue that these are complicated, intricate concepts that the mouse knows, right? You could say, okay, the mouse knows how to count here, or you could just say those are the representations you need to predict the cheese on the 4th lap, right? Because so that distinction between imagining some kind of conscious conceptual knowledge versus just representations, I think, is made even more stark when you think about this task, which is a task I think that maybe Alana will tell you about later as well. But it's a task where the animals have to turn to the left or the right, depending on how many pillars they've seen on the left and the right. And so here in this case, there's more pillars on the left, and so he's going to turn to the left, right? So that task, again, you might think, okay, how are you going to go about solving that task with one of these hippocampal representations? Well, you can, if you know what sensory things have happened in the past. You can turn it into a sequence: left, right, left, right, right, left, right, left. That means I go left. You can just model that in exactly the same way that you model that we've modeled all of the other tasks so far, just predicting your own action rather than predicting the next sensory input. And if you do that, you're going to end up with a little prediction like this, which is that you're going to make a representation that will tile place fields in a 2D space that is not spatial, right? Which is one of these dimensions is the physical position you're on, but the other is this evidence dimension, which is number of pillars that have been on the left minus number of pillars that have been on the right. That's the kind of thing that Tem's going to predict. It's also the kind of thing that David Tank predicted with his lab, and they measured it. And so they measured these place fields in this really wacky, awesome space, thinking that, okay, you've got a dimension for evidence in your brain, which is, or in your hippocampus. So here's one place field, here's a bunch of place fields, and here's Tem predicting exactly the same thing, just by trying to predict the sequences. Cool, so I'm trying to persuade you that, that there's a bunch of things that you can, a bunch of places you can get to, like that seem to be cognitive when all you're doing is abstraction and sequence prediction. Okay, so far I've told you that you can model a few things in the same way, maybe Euclidean geometry, transitive inference. I haven't told you about stereotypical behaviors, but you can do that too. And then some of these tasks, state spaces for reinforcement learning that I showed in the last couple of slides. But all of these have been kind of embedded in space somehow. They've all been rodent tasks embedded in space. What about things that are really non-spatial? And I'm just gonna maybe try to show you, yeah, I've got 15 minutes left. Okay, I'm good. I'm going to try and show you something about now about with humans that are doing sequences, and where there are these kind of abstractions and coordinate spaces built for totally non-spatial sequences. So here's the setup, or at least the first setup. We're going to take patients that are about to undergo epilepsy surgery, and we're going to we've got microwires in the entorhinal cortex and hippocampus, and we're going to record cells whilst they see a very simple sequence, 1, 2, 3, 4, but these sequences are a sequence of images. So it'll go dog, football, key, chili pepper, and then 3 seconds later, we're going to ask them, okay, where was the football, right? And so if they've done that right, they're going to report, okay, the football was at position 2. And we're going to say, okay, well, and the first question we're going to ask is, okay, well, what kinds of representations can we see in this system when they're seeing that? Do we just see representations of the dog or the football and the keys? Or do we see an abstraction, a coordinate system, representation of what it is to be in position 2 or position 4, like the grid cells do in space? All right, so yeah, and in each trial, you have the same stimuli, but they're in a different order. All right. So I'm just going to first show you some cells. These cells are boring, right? Because this is a cell that fires for a picture, the same picture, no matter what position it's in, right? So this cell always fires for the dog, picture 1. This cell always fires for picture 3, no matter what position it's in, the keys here. So those are kind of boring cells. We know those things exist in hippocampus. These cells are a little bit more interesting because these cells are the kind we're looking for. This cell always fires at position 1, no matter what the stimulus is. This cell always fires at position 3, no matter what the stimulus is. And this cell sort of fires anywhere apart from position 1, no matter what the stimulus is, right? So they are building an abstraction of where you are in the sequence, independent of what the stimuli are at that sequence.
Yeah, he wouldn't be a DBS candidate because he lives in Poland.
Okay, so, and the thing to note, one thing to note is that those, it's not just, I haven't just picked out a few of those cells. These abstractions, these abstract cells are equally prominent, explain an equal amount of variance in the, of the data as things that we know exist in the hippocampus, like those picture cells. So it seems like there's some kind of abstraction, some kind of abstract coordinate system for simple sequences, just like there is for for space and for some of those tasks in space. But this is really quite a boring sequence. And so I'm just going to try and take this to a slightly more complicated sequence and see if we can make a richer statement about it, right? So I'm going to have to move to fMRI here. You'll see why in a minute. It's a complicated experiment. So this coordinate system is a more interesting one than the last one I showed you because it's got two levels to it. It's a hierarchical coordinate system. It goes breakfast, dinner, work, sleep, breakfast, dinner, work, sleep, which means this dinner here is at coordinate 2, 3. It's on the second day, and it's the third element of the second day. So we can make that even more complicated by putting a weekend in there, and now we've got a 3-level coordinate system, hierarchical coordinate system, where this guy here is breakfast in the second week on the second day, and it's the first thing, so it's breakfast. Right, so this is now a richer hierarchical coordinate system, but totally non-spatial, right? It's just a sequence, but it's now structured into an interesting hierarchy. So those grid cells that I showed you before have one other thing that I didn't tell you, which is that they exist in one of these hierarchies. They have this beautiful dorsal to ventral gradient where high frequency, short spatial scale grids are in the dorsal portion, and low frequency ones are in the ventral portion. If we were to find that in a human, it would look a little bit different because the human brain is kind of twisted around like this, right? So it would be going more like an AP direction, but you'd still see this fine scale posteriorly and large scale anteriorly. So we're going to look for this kind of gradient, but now in sequences, not in space, right? And so this is how we're going to do it. We're going to take one of these hierarchical sequences, which is just like that breakfast, work, eat, dinner sequence before, but now it's tones, not events in the day. So this is the structure of this sequence. Is an auditory sequence with 100 and something elements in it, and it goes like this. It goes do 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 do, and then the weekend do 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 again and again and again. So it has that exact same structure to it. It in fact has 4 levels of the hierarchy in this sequence. We actually taught them 2 sequences. Actually, don't need to know. Do you need to know that? No, you probably don't need to know that, so ignore that. But the question is going to be, is there this hierarchical representation of these sequences? And we're going to ask that question by doing something, by playing a trick that people play in fMRI, which is we're going to pair time points in these sequences with particular images. So we're going to show them these sequences again. So they've spent something like 2 days listening to these sequences at home, and then we're going to bring them back into the lab, and we're going to show them these sequences again another 25 times. But this time we're going to pair some images with these sequences. And so if we've done this right, then this aeroplane, for example, will be at position 1, 2, 3, 2. And we're going to now go into an fMRI scanner, and we're just going to show them these images in a random order. And we're going to ask, does the correlation of is there anywhere in the brain where the correlation between voxels for different images looks like the difference in the distances at the different hierarchical layers? You see what I mean? So I can look for the similarity in the representation of these things here, and they might look like the things that are have both got a 1 here might look similar to each other, and things that have both got a 2 here might look similar to each other somewhere else. Yeah.
I'm sorry to interrupt you, but I think there may have been a slight miscommunication about the duration of your talk, and it's such fascinating work. But
I think I've got 5 minutes left. No, is that not right? 20.
I think that really, actually, if you could try to wrap up in 2 minutes, so we could take a little bit of a few questions. That was supposed to include the question period.
I was told it was 40 minutes. No, okay, fine.
That was 30 plus 10, but I
Oh.
don't want to waste any more time on this.
Okay.
Just please.
I'm just gonna show you. Yeah. Okay. Fine.
Show us the punchline of that study. I think that's important, and then wrap up for us if you can.
Gotcha. I will show you 2 punchlines.
Great.
That's it. So punchline 1 is that those things are. This is the entorhinal cortex in a human for sequences showing a beautiful hierarchy, just like in the grid cells, but now in sequences that are totally non-spatial. And then punchline 2 is that these abstraction cells exist in this mouse task here. So here's some cells that just care about B, no matter where B is in space, just like the position cells did in the mice. And you don't need to see the rest of this. And then that's the end of me. Sorry.